welcome back. So now we are going to have four invited short research talks. And our first speaker is Heather Beasley, PhD from Vanderbilt University. And she is a postdoctoral fellow in the laboratory of a friend of the Nabrit conference, Antonin Antenter. So it's really nice to have yet another representative of Antenter's lab here with us. And so, Heather, thank you.
Thanks, Mark. I appreciate it. Hi, everyone. How do I follow up after that, right? Okay, we'll try and work with it. But thank you so much to the organizers. I'm super excited to talk a little bit about one of my funded projects through the Bureau's Welcome Fund. And so, as you can see, the name of my talk is Solute Carrier 25846 as a novel mitochondrial regulator and biomarker in breast cancer. So, obviously, we'll be talking about breast cancer. Before we get started, though, believe it or not, all of these little cute, pretty colors are mitochondria. Okay, so not your textbook. So hopefully you'll appreciate a little bit more about mitochondria in 3D through this talk. So let's talk very briefly about breast cancer because we do have just 12 minutes to cover everything. I'm sure you're very familiar that breast cancer unfortunately affects about 1 out of 8 women will develop invasive breast cancer. Now it's denoted by the molecular markers on the outside. So on the outside of the tumor, it'll be estrogen, progesterone, and HER2. But what I want to talk a little bit about are the clinical molecular subtypes. So, hormone receptor positive has the best prognosis for patients. Most patients, about 90%, actually have a 5-year survival, so it's really good for treatment options. The next group will be, of course, your HER2 positive, a much smaller molecular subtype of breast cancer, makes up about 15 to 20% of all diagnosed cases. And what I did my graduate school work on, and what I'm working on also now, is triple negative breast cancer. Now, clinicians and scientists aren't very clever. It's just because it's negative for those three. So, because it's negative for those three, it has, of course, fewer therapeutic options for this particular molecular subtype. And, of course, it disproportionately affects Black and Latino women. And so that becomes a problem for treatment options. So, binding precedes action in science. Since we have nothing to bind to, we have fewer therapeutic options. So now let's talk a little bit about what you probably have already know at some point, which is that mitochondria are essential for cancer development and progression. So of course, it's really important for cell viability. And so that's the problem with cancer cells. They're just cells that are growing out of whack with no breaks. And what we're really interested in is, of course, mitochondrial metabolism. I won't be talking about that part of it in the talk for today, but I will be talking mostly about mitochondrial structural changes. And what I mean by that is that I'm going to show you some images of mitochondria in 3D. And mitochondria actually have 8 different distinct shapes that we never learned in our textbooks that I hope that you'll appreciate even in your own research. Now, let's get started with what we're talking about. So, this is an uncharacterized protein, meaning there's no crystal structure on it, less than 52 papers in publication. Solute carrier 25846. Now, how we found this protein is literally we threw some things at the wall and saw what stuck. Okay. So, one of the opportunities that I'll talk to you about is something at Vanderbilt Genetics Institute, which is how we kind of came up with this particular protein to study. But what I want to show you about is just very briefly what it is. So it's a carrier. As you can see from this picture, it is on, there we go, on the outer mitochondrial membrane. So it's considered a peculiar protein within its family. So the SLC25 family has about 53 different proteins, but this one's on the outer membrane. And because it's on the outer membrane of the mitochondria, it's actually facilitating solutes, mainly lipids, between the endoplasmic reticulum and the mitochondria. Now you'll see a lot of other different proteins that it interacts with, which is why when you overexpress it, it's going to cause some fragmentation. And when you knock it down, you're going to see some hyperfusion or more mitochondria, which is kind of part of the dynamics. So obviously, this is important for mitochondrial fission and also the transport of lipids, mainly phospholipids is what the literature shows. And of course, it interacts with these proteins I just showed. Now, what are the goals of this research? I'll be showing you 2 specific arms. So we know mitochondria drive tumor progression. First, we'll talk about the computational genetic approaches. And the second part is going to be the 3D reconstruction of the mitochondria and what we think that implicates in disease progression. So very briefly, just so you know the rationale for why we're actually looking at this, uncharacterized protein, we know there's limited therapeutic options for breast cancer, right? Especially triple negative breast cancer. Now, what we don't know is kind of some of the ancestral related underpinnings. So, really, how does ancestry play a role in patients developing this molecular subtype? Because it particularly affects, as I mentioned, Black and Latino women. Now that we know a little bit about ancestry and breast cancer prognosis, we want to be able to characterize how ancestry affects breast cancer development and, of course, progression to late end stages. So, the first specific aim, or the first part of this talk, is we're going to look at the association of SLC 25A46 with abnormal breast phenotypes. Now, this is going to be a computational genetic approach using electronic health records. So, basically, if you go into the clinic, you're going to get ICD 9 and 10 code. That is what someone put in. Patient presents with a cough, patient diagnosed with COVID, whatever that code is, it goes into this system. Now, Vanderbilt collects all of that and also collects any leftover information at the patient's discretion. So, any urine samples, fecal samples, blood samples, and we have this biorepository that we can use for future use. And so that's what this first part of this study is going to be interrogating. So these biobanks, the way that Vanderbilt Genetic Institute works is it has a synthetic derivative. So we don't actually know who the patient is. They're giving a special number, but we have over 3 million individuals that have donated to Vanderbilt since the 1990s. Now, what's left over, as I mentioned, any of those samples, we have is much higher than 270,000 now, but we have quite a few of those, and we have them stratified based off of ancestry. The main two groups are going to be European descent and African descent. We actually have over 15,000 patient samples of African descent at Vanderbilt, which doesn't seem like a lot, but it is quite a bit for us to use for these type of analyses. Now, these genotype samples we then can use for other parts of the project. So let's dive into the data. So if you're not familiar with GTEx, GTEx is a genotype tissue expression project through the Broad Institute. Basically, you can go to gtexportal.org, you can type in any gene or protein of interest, and it'll tell you the basal expression across different tissues of different healthy individuals. So you can say, hey, what is SLC's expression in the brain? Okay, this is its expression versus the liver versus another tissue. Now, what we do is we actually model this information or make a specialized computational effort modeling Vanderbilt's BioView, so all of our patient samples, basing it off of healthy RNA-seq value in order to do predicted expression. So now we're predicting expression of patients developing a disease based off of what we have in our biorepository and what we have against healthy individuals. And so what we find is that when we look across 85,000 patient samples, we find that SOC 25A46 is highly associated with breast cancer phenotypes. So let's go into the data. Actually, this is supposed to be first. Here we go. So this is a phenome-wide association study, or a PheWAS. So this Manhattan plot is basically showing us what, just to orient you if you're not familiar, down here are tissues. And then, of course, in the y-axis is just the negative log p-value. Now, this dashed line is going to be anything that passes 0.05, so significant. Upward arrow means increased likelihood of seeing that disease. Downward arrow is decrease. So I'm going to zoom in so you can read the top stuff because I know it's kind of hard to see. So patients that have high expression of SOC25A46 are predicted to have lumps or mass in the breast or abnormal findings on a mammogram. And so that was the first thing where I said we threw something at the wall to see if it stuck. We said, wait, this gene is particularly associated with breast cancer in some form or fashion. Let's see if we can interrogate this data a little bit more. So let me go back a slide. This is what's supposed to be this next one. What we wanted to see is then what is the basal expression differences when we stratify, of course, based off of ancestry. As I mentioned, we're really trying to kind of understand how ancestry plays a role in how this disease is progressing. Now, what you'll see here is that there's very distinct differences between certain tissue types. For instance, for adipose tissue, you can see that in Europeans represented in teal versus the African ancestry patients represented in red, there's very stark differences in predicted expression in that particular tissue. So we're not only looking at tissue-specific differences so we can better understand how we can do the molecular work to tease this apart, but we're also interested that African Americans and Europeans typically have very distinct differences in how they express this particular gene of interest. So now let's look into what this data shows in a huge international cohort. So our collaborators at Melissa Davis, who is just left Cornell, now she's at Morehouse School of Medicine. She's a geneticist that published the front of Cancer Discovery, where they now did an international cohort of samples of patients with triple negative breast cancer. It's the largest sample that they've shown, and they're doing Eastern Africans versus Western Africans because patients, unfortunately, that are West African typically have poor response and have more likelihood of having triple negative breast cancer, just to provide you with some context. But what we find with our study is when we probe within their RNA-seq values, SLC 25A46 has significantly higher expression among self reported African Americans when compared to Ethiopian, which is going to be East Africa, or when we look at the Ghanaians, which is going to be West Africa. So Ghanaians is found here. Self reported African Americans is higher, and then we have Africans or Ethiopians represented here. Now, at first, my first thing was okay, this has something to do with African ancestry. But what's, I guess, more important to understand is, as I mentioned with Vanderbilt's biorepository, 70,000 of those 85,000 that I showed you in the FIWAS are actually European. So now we're thinking it's something to do with admixture, which means not just African that's driving this gene. It's when you have these additions of other ancestral relating things that actually lead to more of the phenotypes we see. So we did spatial transcriptomics to kind of verify this as well. This is with John Carpton's group at University of Southern California. Again, this is triple negative breast cancer patient sample. This is just one reading. We find a higher expression represented in blue of patients of African American descent versus the Caucasian patients. And this is just going to be a readout of where we see higher expression within these two tissues. Now, here's the last part of what we're going to show you very quickly because I don't want to take all your time. This is a really cool technique that I think, is the video going to play? Let's see here. One of the main bread and butters of our lab is we're interested in a technique known as serial block face scanning electron microscopy, or SBF-SEM for short. And so what you can appreciate, just pay attention to the video on the right as I talk a little bit about it. But you can take any tissue, you put it, of course, in this microscope. I won't go through the method in detail, but we do have a published paper right here. But we can actually reconstruct the mitochondria and ER in 3D. And what that gives us is a metric to not only quantify how wide the mitochondria are, what their length is, but also establish contact sites. Now, of course, that's important for this research because SLC is on the outer mitochondrial membrane. Therefore, it is interacting with the mitochondria in the ER. And so the blue is representing the ER and the red is the mitochondria. We find that these contact sites are very important. And so now we're looking at what happens when you downregulate or knock down SLC for what's happening. So lastly, SLC 25A46 is highly expressed in triple negative breast cancer patient-derived xenograft models. And so we're taking that into account. So now we just have a couple of videos just so you can see what the mitochondria look like in 3D. But we find that there's a lot of association within triple negative breast cancer specifically for this gene of interest. Thank you guys so much.
One or two questions for Heather?
Questions?
No?
Do I need a microphone?
Yes.
Hello, I'm China from Brown. Great talk. It was
Thank you.
very, very interesting. I was wondering if you stratified the African American population based off of demographics, like socioeconomic status, access to healthcare, that sort of thing, and see if there are any differences that associate.
That is a great question. We do have, we can actually peruse the patient's records a little bit deeper. One of the good things about Vanderbilt is a little bit of more of an unpopular, unhealthy population. So because we're in the deep South, honey, unlike our deep fried chicken, we're going to have a little bit of differences in that community. But most of the people that are coming to Vanderbilt are coming from a nearby state. So you got Alabama, Mississippi. So what do you think? You know, we're going to have more of a different population. For instance, we're now comparing it to one of our collaborators at Penn who has more of a heart-related phenotype paper. So we're doing that. We can do it, of course, stratify by age and other small socioeconomics, but typically we do have we know a median BMI, we know the age, and we know those factors, and we can take that into account. Great question.
Not sure how this thing works, man.
Thank
Okay,
you.
great talk.
I'll
I
play the
have a
videos while we talk. Sorry,
oh yeah, for sure.
so y'all can just see those.
I have a quick question about the associations that you found because you showed us the African population compared to the European population. And if I remember correctly, I'm assuming that those are patients that showed breast cancer. But do you also find similar association when you look at healthy African individuals, or do they look more like Europeans? And on top of that, I'm a man and don't know if breast cancer affect men that much, but I know it affect mainly women. But if you look primarily in the population of men, uh, that are maybe have other type of cancer like prostate cancer, do you
Yeah.
find similar type of associations?
Excellent question. So, for the first one that had the red and green graphs or turquoise, those were all patients with predicted expression. So, it is predicting it based off of GTEx, which would have been healthy individuals. However, with the graphs that had the plots, those were, of course, patients that had breast cancer. And so, you did see a distinct difference. We would still see probably differences between European and African American just based off of our analyses and other things so far. But we haven't looked just in the male population. That's a great question. Although men don't develop breast cancer as much, they still do develop breast cancer and they could be carriers for their daughters, right? So we still have that genetic link there, but we haven't looked at men. So maybe we need to talk about that. I think that's a very great question because those are things that we don't think about as far as passing the gene along and things of that nature. As far as prostate cancer, there's a lot of similarities between breast cancer and prostate cancer, like a lot. Like even their preferred organ, like is even the bone, just similar to breast cancer. So we probably could do some cool studies and look at prostate cancer as well, which I'm interested in health disparity research. So I'm equally interested in prostate cancer, but just haven't had the time to look at it quite yet. But great question.
Quick question.
Yes.
Did I see on your one of your slides earlier? Is it beta sheet when it's highly expressed, the protein SLC, or did I interpret that wrong?
Sorry, what was your question? You said beta sheet?
Yeah. Well, so I walked in kind of at the beginning.
Sorry,
when you're
I'll go
talking.
back. Which one?
Yeah. Keep going back. Sorry. Right there.
Uh-huh.
So is it normally a beta sheet protein? Is that normally its structure? Is that
Yeah,
or is
as a
that
transporter.
abnormal, or
No, it's a transporter. So it looks just like that. And yeah. So it almost, yeah, beta sheet, GPCR kind of looking, 7-transmembrane. Yes.
Okay, so then a follow-up question real fast.
Sure.
So P53 is like mutated in half of all cancer,
Cancers.
right? So the
Mhm.
triple negative breast cancer, do you find those mutations
Yes.
also?
Most of them are mutated, especially in the in vitro with the cell lines. About more than 50% of them have a P53 mutation. But in TMBC, the main mutation is EGFR, epidermal growth factor receptor. So that's why there's a lot of treatments in that field. But P53 is almost spot on. Yeah, great question. Sorry.
Thank
Thank
you.
you.
Oh, here. Yeah, he's here. Okay, great. All right. Our next speaker is Eric Guillen from Albert Einstein College of Medicine. Eric is an MD PhD student in their medical scientist training program. And yeah, it's a really great pleasure to have you here, Eric, and looking forward to hearing about your work. Thanks.
All right. So thank you everybody for being here, and thank you for the conference organizers for inviting me to talk and also for creating such a wonderful, welcoming environment here. So I'm going to be talking about my PhD project today. So the title of my talk is Exploring the impact of destabilized T-cell receptor signaling on memory-mediated T-cell functional programming. So as you can tell, it's an immunology-focused project. So to get started with a little bit of background on immunology, the immune system is the soldiers that help us fight infection. I'm focusing on one particular type of lymphocyte called a CD8 T cell, also known as a cytolytic T cell. So the main role that they have is to detect intracellular antigens that can help them detect pathogen-infected or tumorigenic cells, so they can help protect the host. So to get a little bit more background, when it comes to vaccine development, a lot of our vaccines focus on developing a very strong antibody response. However, few have focused exclusively on developing a long-lived CD8 T cell response. So there is a big need to understand better how memory CD8 T cell memory takes place. When it comes to activating a CD8 T cell, you have three particular signals that are very important. The first one is cognate antigen recognition. So here I'm showing a naive T cell with this T cell receptor. The T cell receptor, think of it as the eye for the T cell. So it can recognize antigen that is bound on a loading dock called an MHC molecule on an antigen presenting cell. So this is considered signal number one. And that's the signal that I'm most interested in. What happens when you modulate signal number one? How does that influence the eventual memory cell that can form weeks, months in advance? So the other two signals are co-stimulation. So here's a canonical CD28, CD80 co-stimulation. That's very important to further activate the T cell. And cytokine signaling, which can skew whether a CD8 T cell becomes an effector cell or a memory cell, what kind of functions can it carry out? So the big question is, how do these signals, signal 1, 2, and 3, modulate the functional features of resulting memory CD8 T cells? And again, I'm focusing mostly on signal number 1 for the purpose of my talk today. So to get started with my model a little bit more, I'm going to talk a little bit about work that we'd already published in my lab last year, in which we showed that TCR signaling strength determines the dependency on robust cytokine signaling to form functional memory CD8 T cells. In the sense that if you have a context where you have strong TCR signaling, even in the absence of robust cytokine signaling, you can still form very functional memory CD8 T cells. However, in the case when you have weakened TCR signaling, it is required to have much stronger, in this case, IL-2 signaling to develop a functional pool of memory CD8 T cells, which at the end of the day highlights the importance for epitope selection in vaccine design, which is critically important to develop more robust vaccination strategies. So here I'm just showing like a little summary of that work in which we show that cytokine signaling controls primarily homeostatic processes. TCR signaling is involved more in differentiation and metabolism, but the two signals together are very important to synergize and develop the memory program as a whole for the memory CD8 T cell, involved in stemness, cell cycling, calcium signaling, etc. So, shifting gears now to my current PhD project, I'm working with the hypothesis that altering the quality of TCR signaling, which you can define by a few different parameters, in my case on strength and stability, I can produce functionally distinct memory CD8 T cells. The approach that I'm using is I'm using a lot of mouse models. Particularly, we're using a transgenic TCR model. And you and I, we have millions and millions of clones of T cells, each recognizing a different immunogenic peptide. However, in this particular transgenic model, all of the CD8 T cells have only one receptor against one model epitope. This is the ovalbumin-derived OVA peptide. Ovalbumin is a chicken-like protein. So I'm using a series of high-dimensional spectral flow cytometry, epigenetic profiling, and adaptive self-transfers to assess what happens when I change that particular OVA peptide to give a distinct TCR signal. How does that develop a distinct memories program in my CD8 T cells? So to get a little bit more into the nitty-gritty of my system, again, I'm working with a transgenic OT1 system, which, again, reacts to the OVA peptide. So I'm asking the question, so just to get everybody acquainted a little bit, we're going to switch into structural biology for a second. Here, I'm showing an MHC molecule. So again, this is the loading dock, which presents the antigen to the T cell. So, and then I show this crystal structure in which the OVA peptide is bound to the MHC molecule. So my question is, what happens if I mutate this particular peptide? As you can see, there are a few residues that are pointing outward. Think of that's pointing towards the TCR, so they're TCR contact residues. And I have a few residues that are pointing downward, which is allowing the antigen to bind to the loading dock. What happens when I mutate these two particular residues? How is that going to change my memory program? And particularly, if I mutate this position 4, I'm going to generate something that I term weak but stable signaling, because I'm not affecting the binding with the MHC molecule. If I mutate my residue at the position 8, I am weakening my signal and destabilizing the interaction. So I have two different types of mutants. And that's what I'm going to be working with. So whenever you see blue, think of strong and stable. This is what I'm terming my natural signaling. Whenever you see green, think of weak but still stable. And when you see red, think of unstable signaling. So how does that change the memory program down the road? So in order to ask this question, I am immunizing my T cells in a mouse system with three different peptides. And first, I wanted to show that in fact these are actually stable or unstable interactions. So I performed this particular stabilization assay is working with a TAP deficient cell line. TAP is a transporter that's associated with antigen presentation. So in this cell line, antigen cannot be endogenously processed. So you will only see MHC. In this case, I'm working with a H2KB MHC. It will only be expressed at the cell surface of my cells if the peptide is very highly bound to the MHC molecule. What I want to highlight here is that my unstable peptide, shown here in red, requires higher peptide concentration to stabilize MHC on the cell surface, equivalently to my weak and my natural peptides. So that's just showing that this is in fact an unstable signal. To ask the question of how this affects the memory program, I perform an adaptive transfer assay. So I transferred my OT1 cells, which again only react to the OVA peptide, into a wall-type mouse, which I immunized with our model of infection. I'm using an intracellular bacteria called Listeria. And Listeria expresses OVA, either one of my OVA variants, either the natural, the weak, or the unstable. So I immunize my mice, the infection takes place. Eventually, the mouse clears the infection, particularly through the OT1 cells. And at the memory stage, I use fluorescence activated cell sorting to find my memory pool. I transfer a one-to-one memory cell into a new set of mice. So either memory cells that were induced by the natural signal, by the weak signal, or the unstable signal, transfer them to a new mouse, and then I challenge again with Listeria that expresses OVA. So as when you become reinfected with sickness you were already infected with in the past, your immune cells activate again, expand like crazy, and help control the reinfection. And the interesting part is that when I looked at 6.5 days after infection, so this is the peak of infection in our system, I saw that my weak generated memory cells did not expand any better than my natural. Surprisingly and counterintuitively, the unstable generated memory cells expanded twice as much as my natural. So this came from the previous story that we published, and we were confused as to what was going on. Was this a fluke? Was this a mistake? We have recapitulated this in multiple different settings to suggest that this is actually some physiological phenomena that we're recapitulating. So now the question is, why is that the case? Why are my unstable memory cells expanding a lot better than my naturally generated memory cells? And to get to that question, I'm using something called spectral flow cytometry. So flow cytometry is a single cell system used to measure protein quantities in your cells. Historically speaking, you've been limited to about 8 to 10 markers per analysis. With spectral flow cytometry, which has arose more in recent years, in recent decade, I can look up to upwards of 30 markers at one time. So using the same adaptive transfer assay that I mentioned in the past, transferring my CD8 T cells that respond to OVA, immunizing with Listeria that expresses either the natural, the weak, or the unstable OVA variants. And then look, instead of doing a re-challenge, I'm just looking at the resting memory cells. What does that memory program look like? And what that suggests is that, so here I show my preliminary data. Here I want to focus more on the quantitative. I show that first of all, I would like you to appreciate that between the blue and the red, there are stark differences. I'm looking at a series of different markers known to be important for fate determination in T cells. And as you can see, the unstable generated memory cells look very different from the natural. And if you look at the green looks more like the natural. So that kind of like backs up the data that I saw in the re-expansion assay in the slide before. So looking at a series of different markers, so I'm looking at fate extracellular markers, but also transcriptional regulators. These all point towards the direction that the unstable priming condition generates a more central-like or stem cell-like memory program. So now we're starting to create this idea that we have very proliferative cells that have more of a stem-like resting memory program. So we want to explore that a little bit further. And to do that, we're using a separate technique using attack sequencing. So this assay allows us to assess the accessibility of different chromatin. And what we showed here is that there was a set of open chromatin regions that were only accessible with the unstable priming that linked to a lot of stemness-related transcriptional regulators. Again, further painting the picture that unstable priming is associated with stemness. To further push that idea, we performed this more complex serial adaptive transfer assay in which I take my OT1 cells and serially challenge them with Listeria that express the natural, the weak, or the unstable signals, and I keep sorting them, retransferring them, challenging, and retransferring them. Think of this like a serial, like a boosting. Like, with COVID shots, we get the booster shot every couple of months, right? And what that suggested to us is that the resulting memory cells I use a dimensional reduction algorithm using my 30 color panel to put it onto the two-dimensional sphere. I showed that the branching structure is more diverse in my unstable condition as quantified by the Shannon Diversity Index. Another readout for stemness is how diverse are your progeny. So this is further supporting the idea that we have more stem-like memory cells being produced. For the sake of time, I'm going to skip through a few of my slides. Sorry about that, I just want to get to the conclusions very well. So the conclusion is that epitopes that provide a more unstable TCR signaling produce memory cells that are more stem-like, which in our case could be very beneficial for vaccine design because we want memory cells that are very potent, very long-lived. So this kind of like shifts the paradigm of vaccine development a little bit. Particularly, naive CD8 T cells that were primed with an unstable epitope produced memory cells that expand better upon antigen re-encounter, express a more central-like memory, stem cell-like memory phenotype, increase chromatin accessibility in stemness-associated genes. I maintain greater progeny diversity upon serial challenges, and lastly, expand a subset of highly proliferative stem cell memory cells, which I skipped over a little bit. Apologies for that. So that brings me to this working model in which a strong epitope or a natural epitope provides results in memory cells that are very standard. However, when you weaken the interaction between the peptide and the MHC molecule, you produce memory cells that are more stem-like, which we think we can apply this knowledge into vaccine design to make more persistent, more long-lived memory cells. I'd like to thank my lab and our funding sources, and I'll take any questions.
Questions for Eric? Art.
Art Solomon from Brown. It's a fantastic talk. So do you have a sense for, you have a difference at the receptor, you have a difference in transcription, what's going on in the circuitry in between?
Yes.
Do you have a prediction or a guess?
That is one of the questions we've been trying to answer. Mostly I've been focusing on how do you get to that program. And the way that I've looked at it was through RNA sequencing upon priming. So yeah, like I show in this particular slide, I showed at the resting memory state what it looks like. Sorry, let me go back, look at it. Yeah, so I looked at the resting memory state. I've also done like RNA sequencing 3 days after activation, and I showed that the unstable signal gives a widely different program compared to the stable, strong, and weak signals, which seems to suggest that there are some transcriptional regulators at that stage that we think are the pioneer factors that set the program later down the road. But we're still looking into like what exactly is going on at that stage. We think it's happening earlier on.
Noe from Brown. That was a wonderful talk. So typically in vaccinology, or from my experience, we tend to use full-length peptides, in particular for viral infections, and for the design of SARS-CoV-2 vaccines, we use like full-length peptides. How do you think you can sort of make those viral peptides, quote-unquote, unstable to sort of enhance that effect of CD8 T cells?
Yes, I think it's going to have to go back. We have to rely on our structural biology, see how those full-length peptides fit into our loading dock, into our MHC molecule, and then see which particular anchoring residues are present there. So I would propose, for example, for like a COVID design, vaccine design, what if we design like a spike protein mRNA that mutates the anchor residue? Would that make better memory cells? I don't know. It's a hypothesis right now, right? But good question. Thank you.
Hi, I'm Chanel from Brown. Great talk. I just had a quick question. Did you, in any of your work, show, how MHC might be stabilized presenting these antigens? So is it possible that there are more variations of MHC molecules that are presenting this unstable peptide to more T cells so that more TCRs are able to recognize and then induce the response?
Yeah, that's a wonderful question because, there's a lot of different haplotypes for MHC. It is known for this particular ovalbumin-derived peptide that only the H2KB is able to present that stably. One thing we haven't looked at is, are any other MHC haplotypes better at stabilizing what we think is an unstable epitope? Then that would be a different question. It's not necessarily the instability, but rather the different MHC haplotype that's presenting it. That's something that I'm going to look into. Thank you for bringing it up.
Along those lines, you mentioned you were using a mouse line, and that mouse line only has one type of CD8 T cells, I believe. Then that should increase the probability that they bind to your unstable factor. What do you think it would look like in a more native environment where the number of T cells that have that specific one will be very low? And on top of that, have you tried delivering the your peptide without using listeria? Because listeria as a pathogen could also be doing a lot of things.
Great questions. That's exactly what I'm working on right now, actually. To your second question, before I forget, we are testing a different vaccination strategy, one that's a free peptide Trivax. I'm not sure if anybody has heard of it, in which you're giving the free peptide in conjunction with different inflammatory signals. In that case, yeah, we don't have any particular pathogen that's being associated with it, and we know that the vaccination that produces the memory cells is solely by the free peptide alone. So we are trying to recapitulate those studies right now. Still very early stages, and so I can't really give you a clear conclusion on that. So your first question, another thing, yeah, it's the limitation of a lot of the work that I presented today. It's working with the OT1 transgenic system. We are trying to expand that to a polyclonal system in mice as well. One particular study that I've done right now is by serially challenging, in a non-OT1 transferred system, serially challenging with the unstable peptide compared to the stable, either weak or strong peptides. And it still seems to suggest that we're creating a more stem-like feature based on resting memory phenotype alone, but we haven't recapitulated all the other readouts on top of that. But I think that's something we're going to have to do in the next couple of years to be able to extend our findings. Good questions. Thank you.
Thanks, Eric. Okay. Our next speaker is Letitia Franklin from Penn State. Letitia very recently completed her PhD working with Wendy Hannah Rose, and we're really glad to have Letitia here and looking forward to hearing about your work.
Thank you, and thank you to the organizers for putting this together and providing the opportunity for me to come, as well as all of you in the audience. Shout out to my family who came to see me as well. So I'm going to be talking about modeling purine nucleotide cycle disorders using C. elegans. And so I just want to start off by talking about metabolism, specifically purine metabolism. It is one of many metabolic processes that allows for our body to function as it functions. Purine specifically is important for generating adenine and guanine, which we know are used in making our DNA, as well as maintaining that ATP, ADP, and AMP ratio for energy metabolism, specifically for things like walking, talking, and in my case, doing yoga. The purine nucleotide cycle is situated in the center of purine metabolism, and it consists of the enzyme ADSL1, which converts IMP to SAMP, ADSL, which converts SAMP to AMP, and then AMPD1, which rounds out the cycle converting AMP back to IMP. This cycle is important whenever there is increased cell proliferation, increased activity, because it pumps out the AMP as well as other purines that's needed. And so with each of these enzymes, there are rare, or I like to say considered rare, genetic disorders. And I say that because these are very understudied and they're often hard to diagnose unless you know what you're looking for. And so that's part of the reason why I have enjoyed studying these three enzymes. The etiology of the muscle dysfunction that's associated with these disorders is poorly studied and understood. So starting off with distal muscle myopathy 5, it's caused when there are mutations in the ADSL1 enzyme. Symptoms include difficulty walking, running, and heart muscle weakness. To date, there are about 50 people diagnosed, but once again, I think this is an underrepresentation. This disorder is beginning to gain traction. Interestingly enough, it's not even on the NIH website for rare disorders, but the families of these people who have this disorder have started a website. So if you would like to look more into this disorder, you can go to cureADSSL1.org to support their cause. Adenylosuccinate lyase deficiency is more well studied in the literature, and this is actually where the work in my lab started. This disorder is at the intersection of genetic diseases, metabolic disorders, and nervous system diseases. It's caused by mutations in ADSSL1. The symptoms include seizures, delayed motor skills, and cognitive development. And the reason why it's difficult to diagnose is because many of the symptoms resemble those with autism. And there's also facial structure features that have been identified with this disorder, and this has at least 100 people to be diagnosed. And also, in the more severe cases, this disorder is neonatal fatal, and so that's another reason why it's difficult to diagnose. Myoadenylate deaminase deficiency is the most interesting of the three, in my opinion. It's caused by mutations in AMPD1. The symptoms are exercise induced. So, a normal person walking, there's really no initial difference. The best way that I can explain this disorder is an individual with normal AMPD function can go run for 15 minutes, take a 5-minute break, and then run for 15 more minutes. A person with AMPD deficiency could run that first 15 minutes, take that 5-minute break, and not be able to run the next 15 minutes. And so it requires exercise and a physical test in order to diagnose it. You can also get biopsies to diagnose this, but once again, if you don't know what you're looking for, why would you even think to do such a test? So, lucky for us, C. elegans is a great model organism. It has a relatively quick lifespan, which I've shown here. It takes 3 days to go from the egg to the adult, and so my experiments are on day 1 old adults. It's relatively easy to genetically manipulate. We just use RNAi to knock down specific enzymes, and then we run our assays. Purine metabolism is conserved. And the basic muscle unit of the C. elegans is the sarcomere, which is the same as in humans. So here I have a video. Maybe it'll play. Okay. Here I have a video. Can I find the mouse? Oh, it's there. Okay. I think I figured it out. Thank you. Sorry, I don't think it'll play. It's okay. This is what a crawling worm would look like. The important thing here is to note the bends of the animal. That's something that I will come back to later. There's multiple bends within the animal, and then while swimming, there's this one signature bend where they make a C shape, and they flip back and forth between this C shape as they're swimming. So my hypothesis is that purine metabolism supports movement in C. elegans. More specifically, disrupting the purine nucleotide cycle will disrupt movement function. And so I use WormLab, which is a tracking software, to measure crawling and swimming. The parameters that I looked at in crawling is speed, and I'm sorry, I'm not showing the data for reversals, but I happen to also look at reversals as what was mentioned in an earlier talk. And for swimming, I looked at thrashing rate as well as the maximum bending angle. I used maximum bending angle as a way to show the muscle contraction ability of the animals. And so here's a spoiler alert so that if I don't finish, you still know exactly everything you need to know. We know that many inborn errors of purine metabolism result in movement dysfunction in humans. Broadly, we do not know how the PNC supports normal movement, and so my approach was using behavioral and metabolic assays to characterize using loss of function or reduction of function PNC animals. And what I found is that ADSL and ADSS supports movement but does not necessarily regulate the strength of the animals. And there is, in fact, a role for AMPD1 in muscle function. Recall that I said that it's hard to diagnose. And so because of that, in the field, there are some scientists who say, oh, there's no clinical relevancy of AMPD1. And then there are some scientists that say, yes, this is an actual disorder and it is relevant. So I'm on the side of scientists that say that this is relevant. And the final conclusion that I came to is that the pure nucleotide cycle supports neuroregulation of bending angle while moving. So the cycle as a whole has a function. So with the first question being, what is the movement capability? I grew the animals to day one old adults. I either put them in liquid to measure their swimming ability or put them on a clean plate to measure their crawling ability. And I used the Worm Lab software to record their movement. So the first question in thinking about the movement is, are they slower while crawling? So I looked at ADSS1 RNAi animals first, and I saw that there is a significant decrease in these animals compared to the control. This is what we would expect based on the human disorder. For ADSL1 animals, once again, there is a even more significant decrease in the crawling speed compared to control. Once again, this is what I hypothesized based on what we see in the human disorder. And finally, looking at the crawling speed of AMPD1 animals, there is no difference. And I just want to highlight here that I did not exercise these animals first, which is why we don't see any phenotype in their movement right here. So then I looked at the swimming and I used a thrashing assay in order to determine if they had any slower movement. Looking at ADSS1 animals, I did confirm, yes, they thrash slower. Looking at the ADSL1 mutant animals, they are much slower than the control animals, and this supported the hypothesis. And once again, with AMPD animals, a very short bout of swimming does not result in a phenotype as expected. And so I wanted to see, can I get a phenotype out of these AMPD1 animals? And I used a exercise assay. I modified an exercise assay that I found in the literature where I essentially put the animals on a plate. I recorded the first minute of them swimming, made them swim for 90 minutes, recorded the last minute of them swimming, gave them a 15-minute break with food, and then repeated that process. So I gave them 2 rounds of exercise. And what I found is the conclusion is that AMPD1 does support the recovery rate after extended exercise. And so here I'm looking at the wave initiation rate. And as you can see, our control animals and the AMPD animals have the same average wave initiation rate. And at time 0 and at time 90, there is no difference in the AMPD animals. And going on to the second round at time 0, once again, there's no difference between the average wave initiation rate. But then looking at 90 minutes, there's a significant decrease in the AMPD1 animals. And so I also was curious, what are these animals down here that aren't moving? I thought I might have killed them in the process. Turns out that is not what happened, because when I took them out of the liquid and placed them on a plate, they continued crawling. And so they undergo this thing called swimming-induced paralysis, where they have been forced to swim for too long, and so they just paralyze and seize up and stop swimming. And so I thought, as to what degree are the animals bending? I used Worm Lab software to measure this. I'm going to click through really quickly here. Essentially, this is what a smaller bend or a more tight angle looks like, and this is what a more relaxed angle looks like. In the graphs, the more animals have a stronger frequency, the distribution will be towards the right, the weaker frequency would be towards the left. So looking at the PNC animals, I would have expected that ADSL and ADSS would have had a weaker bends because they are moving slower and they have muscle dysfunction. Come to find out, that's not the case. ADSL animals have about the same capacity for bending as control, and the error bars, we use error bars in a very unconventional way in showing that there's a greater distribution in that the animals can actually contract at the same angles at which a normal animal can contract. We saw the same thing with our ADSS animals, except for these animals were a little bit stronger on average, or they chose to bend a little bit stronger on average. And AMPD animals, which we had not seen any phenotypes before at just the initial exercise, they're now showing a phenotype here, which was also very interesting in that maybe we weren't looking at the correct parameters to measure their ability. And so if we spread that data out, this is what that looks like. As I said before, the distributions, it flattens, and then there's this shift to the right. Quickly go through the metabolomics, extracted, ran through LCMS. I just showed a basic schematic of the purine metabolism showing the changes. The blue represents ADSS, red represents ADSL, and orange represents the AMPD. Very overall number of changes in metabolism based off of ANOVA. Not many changes here. No changes in glucose, which was very surprising, and not many changes in the TCA cycle. Overall, this is just showing that the dysfunction looks different for each kind of animal, and the metabolic changes, there were some, but once again, I think flux metabolism would be a little better for analyzing in this case. This is just a repeat from the very first side, and this is my lab. Thank you to everyone. Yeah.
We have time for a question for Letitia. Yes. Dorothy.
So the metabolomics, what you did, was it before the exercise or is it after the stress?
So I did not exercise those animals at all. For the AMPD animals, I know for a fact that the data will, well, not for a fact, but I'm like 90% sure that the data will look different after they've been exercised because they just have no reason to show a phenotype.
Thank you. I think we should move on to the last speaker of the section. Thank you. All right. Our last speaker of this afternoon's session is Chanel Riley, and we have to thank Chanel for sort of, just quickly agreeing to give a talk at the last minute. She is replacing somebody very quickly, so we're really grateful to Chanel. And Chanel is going to take us back to CD8 positive T cells. And Chanel is a graduate student in the MCV graduate program and is doing her thesis work with Laurent Brosset. Thank you, Chanel.
Okay, hi. I just want to thank the organizers for giving me the opportunity to talk, and I do realize that we are running a bit behind schedule, so I'll try to move through this rather quickly. Like Mark said, like Mark said, Eric gave some really great background actually for my talk on CD8 T cells and especially CD8 T cell memory. So I'm going to talk to you today about my thesis work in the Brosset lab, where I'm working to characterize MCMV specific QA1 restricted CD8 T cells. In our lab, we study cytomegalovirus, which is a double-stranded DNA virus, a part of the beta herpes virus family. Over 50% of adults are CMV positive by the time they reach middle age, so it is a very important virus to study. And importantly, there is no vaccine to date for CMV. CMV is species-specific, and murine cytomegalovirus, or MCMV, is a well-characterized model for HCMV. So to bring us back to MHC, as Eric said, MHC molecules present peptide antigens on antigen-presenting cells, or APCs, in the context of pathogenic infection. And they present these to the T cell receptor presented on CD8 positive T cells. Important for my talk, there are two major classes of MHC molecules, 1A and 1B. Some of the differences, MHC 1A tend to be highly polymorphic, whereas 1B tend to have low polymorphism. 1A can bind many peptide ligands, whereas 1B tends to have more preferential peptide ligands to bind. And importantly, MHC 1A restrict or activate classical CD8 T cells, whereas 1B restrict or activate non-classical CD8 T cells. And so the molecule that we're interested in the context of CMV, it says behind here, QA1, which is an MHC class 1B molecule. QA1 is a functional homologue of human HLA-E. Here's a crystal structure of the two proteins overlaid onto one another with the peptide antigen bound. There's about 80% homology between the two. And importantly, like other class 1B molecules, it has deeper binding pockets. So it's very specific with the peptide ligands that it likes to bind. A previous graduate student in the lab showed that without classical MHC 1A present, the QA1 restricted CD8 T cells are important for MCMV survival. I'm going to orient you to this graph. I'd like to point out this survival curve. Here is a mouse group that lacks adaptive immunity, so no B or T cells whatsoever, and you can see that they are unable to survive chronic MCMV infection on their own. However, if you adoptively transfer the memory T cell compartment from this knockout mouse, which contains only non-classical CD8 T cells, those non-classical CD8 T cells are sufficient to protect these mice from MCMV-induced lethality. However, from that non-classical CD8 T cell compartment, if you knock out the QA1 restricted CD8 T cells, about 80% of these immunocompromised mice are no longer able to survive. Therefore, we concluded that the QA1 restricted CD8 T cell population is the non-classical CD8 T cell population that's important for survival of chronic MCMV when there is no classical CD8 T cells present. So this leads me to my thesis work, and one of my major aims is to identify the peptide antigen that is presented by QA1 during MCMV infection. So I have some approaches that I'm going to go through with you as to how I am trying to answer this question. So the first approach I took was doing kind of like a bioinformatic analysis where I quickly scanned the MCMV genome for some peptides that could be predicted to bind QA1. And I used for reference this known QA1 binders, QDM and FL9, which are known to bind QA1 in other contexts. From this, I generated just a list of 9 amino acid length peptides that look similar to QDM and FL9. And then I asked the question whether or not those peptide sequences are predicted to be able to bind QA1 using this database resource tool. Additionally, I also asked the question whether or not those peptides are able to bind HLA-E. And the reason why I did this is because it's well known that many of the peptide ligands that are able to bind QA1 can also bind HLA-E and vice versa. Lastly, I took this list and I ran it through this program called Prosper. So I took the protein that the peptides were from and asked the question, is there likely to be a cleavage sequence within where that peptide lies in the protein? So AKA, is this peptide predicted to actually exist once broken down in the proteasome or the endosome? From this, I generated a list of candidate peptides, and you can see the scores. And so the next obvious question was to ask whether or not these peptides are able to actually bind QA1. So we developed this assay where we take a QA1 presenting cell line and we exogenously pulse with these peptides from the peptide library. And using flow cytometry, we assessed whether or not the QA1 expression on the cell surface might be stabilized. So what we found was that several peptides are able to bind QA1. Here we have a negative M45, which is another MCMV peptide antigen that is known to bind classical MHC during infection, as compared to our known QA1 binders, QDM and FL9. And you can see that these 5 peptides from that peptide candidate list I showed you before are able to bind QA1 at levels similar and the same to the known QA1 binders. And we did find that 2 of our peptides are able to stimulate the CD8 T cells ex vivo, specifically secreting TNF alpha. And again, I have M45 as negative control. And here, FL9, though it is known to bind QA1, is also a negative control because this is outside of the context of CMV. So these are CMV memory CD8 T cells that we're asking the question for. Furthermore, we actually have ordered some peptide tetramers for these 2 MCMV peptides, and I'm currently testing those right now. Next, we wanted to take this into the virus. So we have some MCMV mutants at our disposal, one of which is missing M139, which is one of those 2 peptides that were positive for the TNF alpha production. And we currently have a long-term infection going with these that we're going to ask some more questions about, whether or not they bind tetramers, etc. Lastly, we also have an MCMV open reading frame library. So we have individual plasmids that encode each of the open reading frames from MCMV. And we are working to transfect our QA1 presenting cell line with those MCMV open reading frames, and then again asking the questions if those proteins processed within the cell will then result in the peptides being presented by QA1 and stimulate a CD8 T cell response as well. So here I've showed you that we've identified several peptides that can stimulate MCMV-specific QA1 restricted CD8 T cells. The next part of my project that I want to talk about briefly is characterizing the T cell receptor repertoire as well as the transcriptional profile of these QA1 restricted CD8 T cells. So very briefly, again, Eric gave some good background on this. The T cell receptor contains this antigen binding site, which is hypervariable. And so our question is, what does this antigen binding site look like that is responding to MCMV? The MCMV peptide presented on QA1. I'm not showing any specific data from the TCR sequencing today, but our bulk RNA, we did two approaches. We did bulk RNA sequencing and some single cell RNA sequencing, including site-seq as well as TCR sequencing. And the bulk RNA sequencing showed us that the beta chain is fixed on these T cells, and our single cell data set also corroborates this. I am going to show you from the single cell RNA sequencing data some of the site-seq data. And here, the big takeaway is that we do find a CD8 T cell cluster that is enriched in infected KBDB mice. So here in black 6, you can see that there are some classical CD8 T cell clusters, 0, 6, and 11, that are not so much present in our KBDB and KBDB-QA1 knockout. But what we're really interested in is if I point out cluster 4 to you, this cluster is not super enriched in the infected black 6, but it is enriched in our KBDB knockout and not quite there in the triple knockout. And so this tells us that this cluster, the CD8 T cell cluster, is the cluster, or are the CD8 T cells that are restricted by QA1 and responding to MCMV infection. Furthermore, the CD8 T cells from infected black 6 and KBDB mice show enriched infector genes such as KLRG1, granzyme A, some interferon gamma as well. And we really look forward to taking our TCR clonotype data and looking at these enriched effector genes and seeing what pathways are activated in our population of interest. So in conclusion, I'm showing you here that a CD8 T cell population that does express effector markers is enriched in the KBDB mice once infected with MCMV. And I'll skip over the project conclusions because it is a repeat. But QA1 and HLA-E are good candidates for vaccines because of their low polymorphism, and also because they are able to bind peptide antigens from one another. And this work really elucidates a mechanism that can be used for host defense as well as under homeostatic conditions. With that, I'd like to thank my lab, past members, present members, funding, etc., and I will take any questions.
Oh, Eric has.
Wonderful talk. CD8 T cells are amazing, I know. My question is, do you think that functionally speaking, the QA1 antigen-specific CD8 T cells have like different functional capacity, protective capacity, compared to more classical antigen-specific CD8 T cells?
So do you mean in terms of response or protecting from lethality?
Yes,
So,
both.
So that first survival graph I showed, it was well known that classical CD8 T cells could protect immunodeficient mice from MCMV-induced lethality, and what our lab showed for the first time was that non-classical compartment alone is sufficient, right? And then if you remove QA1 from there, then 80% of those mice no longer survive. So that suggests that they are functional in the sense of protecting from lethality. Now, in terms of the TCR signaling pathway, the stimulation assay I showed the data from had a lot of TNF production, and then in black 6 at the same time point, we see much more gamma and not quite as much TNF. So it is possible that there are, this is something we're really looking into right now. It is possible that there is some different effector functions that are happening in response to MCMV infection with these cells. Thank you.
I'd like to ask either one of you or both of you, what you think about a recent paper that I read about that commensal bacteria spread on the skin will elucidate systemic CD8 responses to melanoma and to O albumin expression. I don't know if you're familiar with it.
Professor Landy, could you identify yourself for the audience, please?
Sorry.
Introduction.
Oh, sorry. Art Landy Brown.
Um, I am not sure I'm familiar with that paper. I do know that, there's a few researchers that are looking at QA1 restricted CD8 T cells specifically on the skin, or other non-classical restricted CD8 T cells, and also QA1. So the FL9 protein, that's a known QA1 binder, also stimulates a different population of CD8 T cells. This is separate from MCMV, and there's been a lot of work to show the functionality in the gut, especially in relation to the microbiome there in the gut. But that's all I got for that. It's great. It's a great question, though.
Great talk, Chanel. Okay, my question is, so I have a collaborator. I'm actually working on her work right now. I don't study CMV, but she's looking at CMV, the co-infection with HIV. So a lot of patients, I guess, develop it as a comorbidity. My question is, do you think that this vaccine work, I'm assuming you're going to make it to vaccine production, will that be helpful for patients to maybe not have the co-infection? Like, can it work the opposite way? Like, if we help protect patients get CMV, and maybe they're in a population where they're more susceptible to HIV, could it be helpful for that type of opportunistic type of viral load as well?
I would think yes. So the idea, CMV, like I said, 50% of the population has it, but most people never know when they get sick. But it's a big problem for organ donors and other immunocompromised individuals. So if you're able to produce a vaccine that can stimulate a memory CD8 T cell response and the patient never gets sick, then their immune system would be more readily available to respond to HIV. Well, obviously that's a very special case, but yeah, it's a great question. CMV really really is something to think about with comorbidities, because it is the immunocompromised population, people who are sick or immunosuppressed, who get very, very sick. Yeah, great question.
All right, thank you, Chanel. Thanks for
Great.
Yeah.
I want to thank. Oh,
I was just going to thank all four speakers, and also Jess Otis, not only for keeping the time today, but for also all the work that you did in helping put together the conference and especially the poster session. Thank you very much.
It's my very great pleasure to introduce our second panel discussion. And again, I'm just going to briefly introduce our two moderators, and then they will take over the show. So to my right is Miles Bartholomew, whom you've already met at the beginning of the conference. Miles is finishing his third year in the MCB PhD program. He is an alum of Xavier College in New Orleans, and he was also a Leadership Alliance student here at Brown. Are there any Leadership Alliance students in the audience right now? Yay! Okay, it's great to see you. Please, we want to connect with you and introduce you to Miles later. Great. And an HHMI Gilliam Fellow. And then to Miles's right is Amanda Elisa Ruiz, who is fifth year. Miles is third year, so you're fifth year. Fifth year PhD student in the pathobiology program here at Brown, and working on malaria, schistosomiasis. I always do that. I'm so sorry. And Amanda is also an HHMI Gilliam Fellow, and the part that I'm absolutely confident about is that we are co-authors on a paper on learning leadership skills at every stage in your career. And our third co-author is A.J. Hinton, who is Heather Beasley's PI. and also an alum of the Nabrit conference, along with Melanie McReynolds, whose name you saw at the end of Letitia's talk. So with that, I'm going to hand off the mic to Miles and Amanda to take over.
Great, thanks, Allison. Is this on? Yeah, actually. Hello? There it goes. So the idea of this early career research roundtable is to inspire us as scientists to be ourselves and to gain some tools from you all and some experiences from, of course, early stage career scholars striving for scientific excellence while aiming for equity in STEM.
So we have invited several early career scientists, namely graduate students and a postdoctoral research, to share their paths to bringing their authentic selves to the bench, to the lab, to the classroom, to academia, and also to inspire us with some of their insights into starting on a successful career path. So for our panelists, let's start off by sharing your name, your current position, your institution, and your career aspirations.
Hello, everybody. So my name is Eric Guillen. I keep saying third year, but I guess I should say fourth year now. MD PhD student at Einstein. I forgot the rest.
Your career aspirations.
Career aspirations. Yes. So I'm in an MD PhD program. So I think long term, I really want to have my own lab myself. So I'm going to finish my program in a couple of years, then be in residency for a couple of years, and then fellowship. I don't know. We'll see. So yeah, eventually I do want to run my own lab, and particularly I'm really interested in immunology and vaccine design.
Hi, my name is Letitia Franklin. I just defended my dissertation or my PhD work at Penn State. Future career, really, thank y'all. Future career, really now career. I'm really interested in academic leadership, but I also want to still have some type of tie to the bench top. So I am not totally sure. We're going to craft up something and I will keep y'all updated.
Hi, everyone. I'm Heather Beasley. I'm a postdoc at Vanderbilt University. I've been in it for about 2.5 years, but I've been with AJ for over 3 years, so that's been fun. He recruited me from grad school, so he couldn't get rid of me. My research goals is I definitely want to have an independent research lab. Just not sure if it'll be an academic or a governmental institution or industry, but definitely want to run things.
Hi, I'm Sorel Wonka, graduate student here at Brown, finishing my 5th year. Actually, my 5th year is finished. For my career aspirations, immediate career goal is for me to hand in my dissertation and to graduate. I'm among the old ones in the program. Might be time for us to move on. But long-term goal, I need to make money because I just became a dad. But I consider myself to be more of an open scientist, so I don't usually like to project myself way into the future. But if I had to do it, I would say my long term goal is to apply the research that I've shown you here today in different underdeveloped areas in the world, especially in Africa, because I was born in Central Africa before immigrating to the US, and all the things that we see with global warming is affecting those areas even more compared to how it's affecting her in the US. But again, that's way in the future.
Awesome. So, just to start us off, we want to ask, what does authenticity look like for you in your current spaces? That can be, a lab, department, academia as a whole.
All right, well, I'm not shy, so I'll get started. For me, I think authenticity is I consider it a privilege to work with another minority scientist. So my PI is also Black, and so I have the privilege of being my authentic self in a lot of spaces. And one of the arms of our lab isn't just in, of course, the beautiful mitochondrial structures, but also in the arms of DEI and STEM education. And so, with that, we really make sure that we have a very inclusive lab, and that's inclusive of not just race or ancestry, but also inclusive of beliefs, right? Inclusive of religions and anything else that a person holds to be true to themselves. And for us, authenticity is be yourself, show up, be you. You know, if you want to dye your hair purple, we're here for it, right? There's just all these different things that I think we don't see in the typical normal setting that we have in our lab to make sure that we show a different representation of science. And so I strive to carry that on in my own personal life, especially through social media. I'm very active on social media, but I think it's very important for me to redefine for a lot of people. We heard from this second keynote today that representation matters, right? And bringing yourself and showing up as your authentic self can really draw people to you just by who you are. So for me, authenticity is what you see is what you get.
I couldn't agree more. Again, for me, what you see, what you have. If I don't want to smile with you, I will not smile with you. If I don't want to do anything with you, I will not do anything with you. But yeah, for like, I don't consider myself like to be like to do as I'm doing science. Science is part of me, and is one of my passion. I usually tell Mark that if I the day I will stop being passionate about science, that's the day I will stop doing science. So because it's a passion, you have to be yourself to be able to enjoy your passion. So that's why I've always just brought what I am, whether it's here, whether it's at home, because I've also come from a huge family. I have 7 siblings, by the way, and if you don't learn to impose yourself, if you don't learn to be okay with what you are and who you are, uh, you can end up losing yourself and forgetting the things that you love. So being authentic is just taking the science and anything that you do the same way that you would behave as it when you're at home. And if people like it and want to go along with you, great. If they don't like it and don't want to be part of your journey, great too. Not everybody has to like you.
The first thing that came to my mind when you asked that is the fact that I'm openly a yoga instructor. So a lot of people, and I said openly because they, like, it's just thought of like you do things outside of science, like you step out of the lab. And I'm lucky to have a PI that allows me to do pretty much anything I want to do. Like, she has not told me no. She has not told me, I don't think you should do this. I mean, her thing is like, well, do you have the time? But she essentially, for me, authenticity is being able to show up and not hide any part of myself, being able to do yoga, do the diversity, equity, and inclusion work that I do, bring high school students into the lab and mentor them, and to be looked at and accepted and often praised for just doing those things that I love to do. So going back to the passion, yeah, so just being able to do the things that I'm passionate about.
Yeah, and just like I agree with everything everybody has said, and I would just add like authenticity is not always a given. Sometimes it's a conversation with yourself. I think for me, like in the last 3 years, I've been learning to be authentic and finding spaces where I can be authentic. I think like I developed a really great relationship with my mentor, with my lab. As neighboring labs will tell you, we're always like laughing, like just being absurd, and like that's who I am. So like finding a place where you can be that, and like share your aspirations. Like I love like DEI initiatives. I'm part of like the minority scientist organizations on campus, and having a PI that's supportive of that has been very important to me. So just being very honest with what you need, what you want, and a place to for me it means to be goofy. I love being goofy, and if I can bring that to the lab, that's amazing.
Well, thank you for all sharing. As a follow up, what have been some of the biggest challenges to being your authentic self? in academia and in some of these spaces that you take up, whether it be in outreach, whether it be in mentoring, whether it be in an institution. And if you are still working on that, what are some of the things that you actively do to be your authentic selves?
If I may, the biggest challenge has been imposter syndrome. I think that's been like, it's, I don't want to like belittle myself, right? But it's been in my head a lot, and like the thing held me back quite a bit early on, preventing me from putting myself out there, from doing the things that I want, interacting with the people that I want. So I think it was just like a part of the process of building that confidence. I'm still building that confidence. But just the opportunity to like just feel comfortable in like your own skin and your own shoes as a scientist. Yeah.
For me, my biggest challenge has been admitting that I need help sometime. I'm used to be very independent and doing my things by myself and figuring things out by myself, but sometimes that's not enough. Sometimes you need to be able to get out of that comfort zone that you know and be able to tell people, okay, I need help with this, or I need help with that, whether it's academic or personally. Like something that I'm dealing with recently is that I'm starting to see that I'm showing attention deficit disorder characteristics, and for many years, because before getting to this stage, I've been able to handle it very well, but it's it's important to start thinking about when that thing that is happening to you might start to hinder you in where you want to go, and knowing when it's important to step out of that comfort zone and ask people to give you advice or give you ways to even better yourself and become a better authentic version that you will want to be.
So next, I guess, in you guys' experience, how have academic institutions and organizations created a supportive environment where you feel free that you can ask for help or explore your passions like yoga, and how can we encourage other early career scholars to take risk and pursue innovative research ideas that they're truly passionate about and get into projects that they might otherwise be scared of?
Okay, here's a shameless plug coming in 5, 4, 3, 2, 1. So I'm I'm the vice chair for National Black Postdoc Association, and I think that it's a very inclusive environment because it allows for us to connect, especially at these R1 institutions. There are very few Black postdocs at that level, and so finding a community of Black and Brown individuals that look and identify like you, may have similar cultural upbringing, similar values, maybe Greek life, or something that connects you, I think it's been important. Another thing that I would say is I find such a community in a lot of social media. And it sounds great. Social media is not all great, right? I'm not going to paint this picture like it's glorious. There's some really bad sides to it as well, everything in moderation. But what I've seen is through graduate school, I did a lot of advocacy work for patients that had triple negative breast cancer. You saw my talk. I love it, right? And I had patients that would reach out to me in my DMs like, hey girl, tell me about this. I'm really struggling. Like, what does this mean? And it really changed how I think about scientific communication. So they became a support system for me. Like, if I'm saying something and they can understand it, I didn't explain it well, right? And so it adjusted how I communicate to lay audiences, how I communicate to scientific audiences, and how I communicate to people that may be afraid to even go get that mammogram. And so although that's a little bit different than the question, I think that finding community can be in different places. I find it through people that, in the institution, but I also find it through people that I don't even know that actually are rooting for you and wanting you to do well and be supported.
Can you ask that question one more
Yeah,
time?
of course. So, in your experience, how have academic institutions or organizations created a supportive environment that have encouraged you to pursue innovative research ideas or to pursue your true passions in science?
So, I had like so it's not that they're not supportive, but it's more like what I learned along the way is that you have to advocate for yourself. You have to be confident in yourself. You have to go out and go after things for yourself. And then when people see you moving in a certain direction, that's when they'll gravitate towards you to say, oh, if you're interested in this, here's this opportunity. Or if you're interested in that, you could go and try that program and things like that. And I think someone mentioned this earlier, might have been in the keynote speaker session, but like you have to be able to, you will get to a point where you have to start saying no because they start seeing, oh, she's doing this, she can do this, she's doing that, she can do that. And burnout and bound, all of that, you have to pay attention to those things. But just taking those initial steps into a direction that you're interested in. And for me, in my experience, the support has followed that.
In your experience, how has the institution or organizations that you're a part of how have they supported you in these spaces to pursue your true passions?
Okay, so first, personally, I believe that as graduate students, like a keynote speaker said earlier, we've accomplished so much that I believe we can succeed at any institution that we go. And a PhD is about doing your research, and no matter what you do in the research, you're still learning the skill and critical thinking that are necessary to like switch to any field that you want. When I was deciding where to go to grad school, for me, what the most important thing was to make sure that I got into an environment that I would know would support me. With the way that I have. For example, the reason why I selected Brown over many other universities is because I never have to call one of the PIs by doctor. I call I call Alison, Mark, Gary, and so forth. And I never really had. I mean, I knock on offices when I have to go, but that's just because that's what you have to do. But I never felt like any of the PI's door were closed for me. And again, as someone like me that comes into a big family, that comes from a big family, and is used to be surrounded by people, that was something for me, which because coming to grad school was the first time I went away from my family, and I was really scared of being alone that far, and really having that community around helped a lot. And so again, sorry, I forgot your name. Cassandra. So Cassandra mentioned earlier that on top of the work that we do, we have so many other things that we also have to deal with. So it's important to get those people that you know that no matter what, you can just walk into an office and be like, things are not okay. And for me, I know that I have that in Addison, I have that in Mark, and I also have that in many of my older cohort members over here. But on top of that, really, Brown has put together. I mean, there's still a lot of work that needs to be done, but I can acknowledge that here at the university, there is a lot of things that's being done to make sure that we are in the best conditions possible. And in recent years, we have started conversations that we hope will continue to move the needle forward even after we leave the campus. So yeah, I think that's a long answer for your question, but it's not really about what the university has done to help you, but what has the university done to help other people that were there before you, and how is that university going to make you feel the most like yourself. Yeah.
Yeah, again, all of this resonates a lot with me, and I just wanted to add like a little bit about finding those spaces outside of the lab, outside of the classroom, has been very important for me. For me, that means like, for example, if you're familiar with like SACNAS organization or other institute, like I'm part of the Einstein Minority Scientists Association, where I get to interact with a lot of older students, and I think that's been super, super impactful for me. These are people that like have gone through very similar situation not too long ago. They give me really great advice, really good like reassurance, which is really helpful. And even beyond that, like for me, going to like we have like open gym for soccer, and like having like a couple hours a week where I just don't think about anything related to science has given me like a great sense of like comfort and like reassurance, re-energizing myself. Going to institutions, I feel like I have never encountered an institution where I'm like, wow, this institution is perfect. Every institution has its strengths and its weaknesses, so I think also just being able to find those few people that are there to support you, the few people that are going to be able to answer your questions, like very honestly. They're looking out for you for your best interests. I think that's been a skill of finding like those like beyond your PI even like the mentors that are going to be helpful and help you solve problems when they come about because they will come about at one point. Um,
Thank you. So the next question pertains specifically to the title of the panel. So it's starting on a successful track, and we want to know, looking back on your training, what were some critical steps that you took, whether it was going to a specific course, a workshop, a conference, reaching out to specific mentors. What were some of those critical steps that you took to lead you to be successful in your training?
if I may plug mental health for a second, I think somebody, yeah. So I was like undiagnosed generalized anxiety disorder for years. And graduate school was the first time that I actually like gave myself the time to get it looked at. I went to a therapist. Fortunately, my institution has a really cool counseling center, and I was able to like get on medication. And I was like, whoa, like medication helps out a lot. So I would just say give yourself the opportunity. I guess yeah, it's a problem. Not everybody has access to it, but if you do have access to mental health care, there is no shame about it. The first time my therapist pitched medication to me, I was like, no, what is this? And then I got a medication. Here we are, like a year and a half later. And I was like, yeah, like that helped out quite a bit. So give yourself the chance to like, have your needs met. And that could be physical, that could be mental, that could be social, whatever it be.
I love that, and I love that you plugged for that. Adding to thinking about how we take care of ourselves holistically, I think the biggest help for my career thus far is establishing mentorship. I think I had a very skewed view in grad school that your PI was your only mentor, right? And I think that kind of set me not up for success when it came to postdoc because I didn't really know that you could ask other people questions. I kind of had this dynamic in the lab where it was like you ask your PI only. You would ask your committee, but then the PI would go against your committee sometimes and tell you to do certain experiments. So it was very I was only asking the PI, and I that kind of stifled me a little bit. And so it wasn't until I connected with AJ and he was like, "No, mentors, plural. You need a mentorship committee. You need a postdoc committee." What I say doesn't always go, but it goes. That's what he says. But he doesn't mean that, right? I can ask other people in my department, friends, peer mentors, people that I can ask for different advice. And that allowed me to really broaden my reach, but also diversify my thinking. And that was something that really changed the trajectory of my career. It established a lot more collaborations, which led to a lot of currency, which is why I have a lot of papers, right? All of this came together because of the mentorship, which I didn't really have in grad school. Not to say that I didn't have a fantastic mentor, I did, but I didn't know that you were supposed to kind of go beyond the four walls of your lab.
I just want to add on to the mentorship tip. Um, for me, what led to my thriving success was having mentors for every area of my life. So I had what I would call a cultural mentor who kept me connected to my Southern upbringings. I had my scientific mentor, my advisor. I had my professional mentor that was outside of the research, but more so like just professional networking and socializing in the broader science world. And then I also had my fitness, my yoga mentor. So I that really helped me to stay balanced as a student and knowing that I'm more than just the science and that I did not forget who I am, nor did I forget to build on things that I'm passionate about.
That was really great. And I think also since we have a very interesting mix of people in here, if you haven't heard of the Mentoring Mosaic by the Leadership Alliance, it's something that I would definitely like to plug. It's helped. I think a lot of maybe we've all heard of having different people for different things and aspects of your career. So as Letitia just mentioned, like you can have a person that isn't a mentor, even in the sense of like they're telling you advice, but a person that can advocate for you when you're not in the room, or just supply you with things or materials. Not every mentor serves every single function. I think Letitia uh, spoke on that a little bit. For the next question, I would like to hear what or how can early career scholars navigate conservative academic paradigms, like traditional hierarchies, like you mentioned, and how do you challenge those in a way that's respectful and also, serves you as well?
Great question.
I guess my only thing of going against the system at this point is the fact that I was so involved outside of the lab and in the university and teaching yoga and things like that. And I honestly found that my advisor was my biggest support. Like, she was my sponsor, she was my mentor, my academic advisor. So she did wear multiple hats for me. But knowing that she was in my corner and would go to bat for me and would just always advocate for me however I needed was super helpful. So I guess what I would say to build on that is maybe not having one person in your corner and being your sponsor, but having multiple people and also seeking counsel, right? If you are doing something that goes against the grain, talk, don't just do it. Talk to people, get opinions, advice, information. But once again, ultimately, it's still your decision on whatever you choose to do because it's your life. Just knowing there's consequences for everything, whether they're good or bad consequences. But having support, I think that would be the biggest answer to that, or my main answer to that.
I love that answer, and I'll piggyback off of her word of advocacy. I think that's something I also learned in graduate school training that your mentors, your committee, if you're at the graduate level, even your postdoc committee, even your faculty committee, people will speak your names in rooms you aren't in to support you in different positions. But it also comes from building relationships. I think so much as a trainee, we just think that we're just isolated, we're not on the same level, which I love your answer about having a first name basis, being able to walk in when you build these relationships with people, they're like, no, let's look at this a different way. You'll get a different perspective, and it'll really help you when you do have those challenges, because challenges do exist in academia, right? We're not going to act like they don't. But even in a conservative R1 institution, wherever you may be, even when those challenges happen, you will have advocates and mentors that can support you, even if your ideas might be a little bit against the grain. So I think building those relationships will kind of support you, even if you're not kind of doing something that's normal.
I don't think I have a really great answer. I think I'm still working through that as we speak. But I think one way that I'm approaching it, I think, I forgot who brought up the idea of like currency, like with papers, I believe, it was Cassandra. I think that's kind of how I'm approaching it right now, where I'm like trying to build up my foundation, my credibility, and I hope that in years to come, when I feel like I'm in a better position, like I think I can make like better change. That's one way that I'm approaching it, but still trying to figure that out.
Strategy.
It is. It's a good one.
My answer is going to be a little bit different than the rest, even though I agree with everything that you guys said. But for me, the what I've found is that the best way to fight the system is to be the best at what you do. Uh, you're not obviously not always going to be the best at what you do, but you can always strive to be the best at what you do in hope that as other people see you do that, they will be more willing to give a chance to the next person that is coming right after you. And But at the same time, it's definitely very important to be able, even if you cannot make changes, to be able to understand the system and to be able to see how the system works. And I think that's something that I've been lucky enough to see here at Brown. And in the past, for example, someone like me, where I grew up, you don't really have this culture of fighting back or being involved and stuff like that. We usually just do our own things and move on. But also, something that I've been learning here at Brown and that I feel like I should do more of is just learning from other people what they are doing, what issues are having, instead of just being on your side and on your own like that. And in the when we're talking about mentors, I think it's also important to mention that on top of all the mentors for just for that, but when I started at band, I used to think that mentors were usually people that were ahead of you, that were like already had something done, and that they would come and get something from them. But what I've noticed over the year is that a mentor can be anyone. My like for example, I showed you a bioinformatic tool that I developed, and that when I started here at Brown, I had no coding skill, no bioinformatics skills. And I got started on that because there was a student, Kelly Pam, never going to forget her. Right before the pandemic started, we were chilling in a class together, and I just casually mentioned to her that I had this crazy idea about building these high throughput tools, and I had no idea how to do it. And she's the one who basically started pointing me out to online tutorial, because at the time I didn't even know that coding is literally just googling stuff. Uh, but she's basically the one who introduced
Secret.
yeah, she's basically the one who introduced me to that principle, and the rest is just history. So yeah, I think the conclusion to that is anyone has something to teach you if you are willing to listen, and you should. And in grad school, I think what has allowed me to move forward is that even if I know, I don't necessarily need to be the one giving the answer. And it might be a good idea to just let other people speak and hear what they have to say, because what they are saying might not only complete your own idea, but might also open a new area of thinking for you.
Well, Miles and I see you all as being the next leaders of molecular life sciences. And in thinking of the roles that you will play in the future, we were wondering how you think you would promote a more holistic approach to evaluating research impact and contributions to academia and science.
Yeah, I guess that I currently have no idea.
I think I think that is a loaded question. I think, and I'm gonna take it from my personal perspectives because that's how we define a lot of and shape things, right? We take information, we learn more, and so that adds to our information. What I've learned a little bit from being in AJ's lab is you can wear multiple hats and be equally successful in both, right? So my lab published a paper just 2 days ago in Cell about Juneteenth and what Juneteenth means in STEM. And so somehow AJ liked me enough to give me a first author in Cell on Juneteenth. So it was super cool, right? To share my voice, my perspective, what I think it means to STEM. But I say all that to say our lab has had over 10 cell papers just on DEI. That's kind of unheard of to have such a high impact journal really focus on issues that help diversify how we think about science. So, my personal interpretation of how we can look for science differently is holistically looking at the scientists. Cassandra mentioned earlier that only a little piece of these R institutions look anything other than science, right? But I think we can change the narrative a bit where we look at, scientists and their advocacy work. We look at their initiatives to affect their community, and we also look at the science. So for me, my personal goal is to be impactful in many areas, but making sure that it still identifies what I find to be my truths and what can help my community.
We were wondering, in terms of your leadership roles in the future, how do you think you could promote a more holistic approach to evaluating research impact and the contributions to science? So, whether it's focusing more on mentorship, right? Maybe at an institutional level, Leadership Alliance, which is housed at Brown University, has made a very big impact in the realm of bringing in undergraduate students into the research labs over the summer. So, is it maybe something in that regard or maybe something else?
So I initially had a thought about what do the people who give out grants decide what's important. Um, but then I decided no, that's not the direction I want to go in because I still have a lot of thoughts about that. So I think my answer to that question could be allowing the next generation of scientists and really allowing myself to know that what I'm interested in is important, even if it's not deemed as important by the grant um, the grant funders. Thank you. And helping people to realize that, there are, I don't want to say alternative. There's always like, so something that maybe costs $1 million to do research wise, get a little more creative and break it back down to what can you do if you got $1,000, right?
Did you say that?
I'm sorry, it's my authenticity. Sorry, go ahead.
Getting some type of something to show that this is credible, this is worthy of following up on, or just being persistent and not taking no for an answer, but having more support for people to do the things that they're interested in instead of feeling like they have to follow the trend of what's being funded.
Yeah, because our research isn't like hot research, right? Some of these mutations that I look at may affect 1% of the population. It's not going to get funded, right? But if we can find that this technique works across another disease that maybe is like cancer, it's like, yes, they'll fund that, right? So, but it's still not to disregard that 1% of the population still might be affected. So I agree with your point that as we look at science and think about granting or grants in general, grant funding, we do have to not take it so personally if it's not hot science sometimes. Sometimes the little follow ups can lead to great discoveries.
And final question before we get into questions from the audience. Kind of pivoting back to something that was previously mentioned, but how did you guys, y'all are all at different institutions, how did you guys find community that really got you, past grad school or into your final years of grad school, just for the audience?
I would just say older students really got me to where I am. I think like yeah, like established faculty mentors were super helpful, but I don't think I would get to where I am right now without like older students giving me that guidance, putting in that labor for me, answering my questions, revising my essays. Yeah, that's one thing I would say. Like just find those older students that are going to be helpful to you, and the faculty members too, because they're also been very helpful. Like my intro cell bio professor has been one of the most impactful people in my life. Like I cried in her office so many times. So I think like just find those people you can be like your authentic self with. Yeah.
I was a student that read all of my emails. So, and I joined lots of listservs and I just showed up to things. I probably would go to things. I might try to pull other people in, but most of the time I would just show up to something by myself and then make friends there. I also really honed in on the identities that I found very important to me. So I joined Graduate Women in Science and I took leadership roles in that. And then I joined the Black Graduate Student Association and I took leadership roles in that as well.
Grad school was a little difficult. I'm not going to sugarcoat it. I think, honestly, my community wasn't necessarily in the students, but it was mainly in my family. I am not a first gen student. But both my parents are lawyers. They had no idea how to direct me. One was like, "Yeah, be a scientist." I don't know how you do that, but do it, right? I didn't have that kind of direction, but I did have an older sister. She's a DRPH. She's in public health. She kind of helped me navigate academia. And I think that was super helpful. So she was, even though we're 4 years apart, was like a peer mentor to kind of support me in every way through it. She's like, girl, the defense is crazy, but you'll get it. And we walk through those issues. So I think family was my community through grad school and even now to this day. And then I've adopted other mentors that have kind of supported me. But it wasn't necessarily in my peers at that point in time. It was mainly in people that I really trusted to be my authentic self before I, progressed.
So when you're among the older students, you don't really have any older student to go to.
Yeah, me
But-
too. I was an old student.
But in previous years, we each were given mentors that we are still in contact with. For example, I'm Miles' mentor. We don't really talk all the time, but he knows that if he has any issues, he can always come to me, regardless with even in no question asked. But I think there are many communities that we get in grad school and that we should get in order for us to navigate that. I'm already mentioned earlier the university, department community, the fact that we are all informal, we are all scientists, regardless of whether we have 30 years of experience or if you are starting tomorrow. And that's something that Mark taught me earlier on in my career, that I should never treat any of my undergraduate researchers as subordinates, even if they are not skilled yet, they're just scientists in training. So that's very very important. But I also have other communities. For example, I have two people here on campus from my cohort, and here at Brown that I don't know how it is in other universities, but there's really an effort to try to get you close to the other people in your year. And in my cohort, I'm lucky enough to have a few people, especially Azo and Chanel, that I know that I can disappear for months, but the day that I need them, I can just say, "Hey, are you available tomorrow?" And with a no question asked, I will be like, "Okay, I'm there for you." Uh, so on top of that, I have my family. Like I said, I come from a huge family. I have my wife and kids. Every day when it gets hard, uh, I find happiness in knowing that no matter what happened, I'm going home to them. So that makes it a little bit easier to go through the day and to start the next day, because when I get home and I look at my son, sorry, just, when I get home and I look at my son, it just makes everything better. Uh, yeah, and on top of that, we also have the other students in the program. We have a volleyball team that I haven't been to for a while, but we usually play intramurals and we suck, but it's fun. But yeah, it's just finding all those other things that sometimes makes you forget that what we do is hard. I think that's what's important.
Thank you. So it seems like for our panelists, community means finding those near peer mentors, finding those individuals that share your values, finding identity based organizations where you can share and also have those counter spaces where you're able to talk about issues that maybe don't affect everyone in your program, but affect a few, and you can have that bonding going on. And also to think about what currency we do have, whether it's familial ties, whether it's the networks that we have outside of the lab. So thank you. It's really important to share all of that. So I think now we're going to turn it over to the audience, and if anyone has questions.
Great, thank you. Thank you all so much for sharing your wonderful insights. I feel like we talked a lot about kind of community and institutional elements, but I'm curious about the lab environments. Do you have specific recommendations or observations about what builds a good lab environment where you can really be your authentic self?
Uh, so I think the lab environment for me doesn't really have to do with whether we are uh, young scientists or minorities or anything like that. The lab environment for me is whatever environment makes you want to go back there the next day. If if the lab doesn't want to make you wake up in the morning and do work, then maybe it's not the right lab for you. Me, for example, uh, I'm in a lab where I know for a fact that I will have the support that I need, but I'm also in a lab where I know that if I need maybe to go away for a little bit, I can just go away for a little bit. I also know that I'm in a lab where there are people that I can talk to about just more than science, and also in the lab where I have the opportunity to train a lot of scientists, because I started my scientific career in my senior year. Would have never been a scientist if there wasn't that one graduate student, Sean Higgins, again, I never forget these people, that looked at me and was like, "I'm going to give you a shot, even though you're already a senior." And it was after that that I was able to get all the experience that I need to be accepted at an institution like this one. So it's more about whatever makes you feel safe, whatever makes you feel yourself, compared to something having to do with us being minorities, I believe.
I love that. And I will add to that. I think that having an inclusive environment is key in a lot of different ways. I think most people have, even if you haven't personally experienced a toxic work environment, you've seen one on TV or you've been told about it, right? And from my experience, the toxic work environment, as you said, is one you don't want to go back to. Like you dread going in. You go in opposite times of people. You really just like, I'm not culturing these cells till midnight because I know no one will be in there. And that's not really helpful for science in a lot of ways because whenever you stifle creativity and comfortability, you really kind of affect the output. My cells always got contaminated when I didn't want to be around people, right? Because I couldn't, be in an exciting environment. And you talked about wanting to be goofy and being your authentic self. When you can't be, it's not a good recipe for success. Now, does that mean that lab is always going to be rainbows and butterflies? No, it's not, and you have different personalities. But I think one of the key things that's worked in our lab is we have a PI that's very in tune with what every person's doing personally and scientifically. And so it's like, we have moms. I'm one of the moms in the lab, right? And so it's like, are your kids okay? Like, do you need to go home? And I'm like, oh my God, my kid has an ear infection. I can't be here today. It's like, okay, well, you're going to be doing structures for the next week. Just work from home, right? So it becomes an environment that is supportive for where we are, right? And that is why it works. Or I can call our staff scientists and like, girl, I forgot that antibody out. Can you go put it in the refrigerator? Because I live way too far, right? And they're like, okay, let me go run to the lab and do that for you. But it's only when you kind of have an environment where you feel comfortable. I've been in lab environments where you don't even want, like, you're scared that your cells are going to be dead or your mice are going to end up dead because the person you thought was sabotaging you. I really have been in those environments, right? And so I relate to how it must feel when people are in a toxic environment. So that is something that I appreciate, that we don't have an environment where you can't feel safe to not only do your science, but to be yourself.
I agree with all of those things. And the only thing that I would like to add to that is knowing yourself so that you know what type of mentorship that you need from the advisor. So my advisor is extremely hands off. She tells you what you need to do, or or we will converse about what I need to do, and then it's up for me to figure out when I'm going to get it done by whatever date we agree upon. There's some people where they need really hands on and like just yeah, they just really need hands on and direction. I'll just say that. I'm not that person. So my advisor and I, we worked very well together. There's some people where she just did not work well with because they needed more of that hands on.
Yeah, just following that idea, I strongly believe there's no such thing as a good lab or a bad lab. There's just good fits and bad fits. I joined a lab where, when I was talking to like somebody who had rotated through the lab in prior years, I told them that I was thinking of joining this lab. They were like, "Oh, don't do it," and I was like, "Wait, why?" So then I started like getting a little insight, and yeah, like what to one person seemed to be like a micromanaging PI, to me was somebody who was very supportive and very hands on. Like you mentioned, like I needed that hands-on small lab environment. Somebody like I could ask a question like in the moment of doing my experiment, like, wait, do I add this or do I add that? Like I think that's something that I really valued. So I would just say like try to find a lab that is gonna like play to your strengths and support your weaknesses for sure. And then going back to like lab environment, I think for me with me and my PI, I have like kind of like church and state, right? Like I to him, I talk about science and science alone, and I like that. And then with my lab, I am like like I said, like super goofy or just fooling around. Like I feel like I'm on like The Office, like the workplace type of shows. Like I feel like we're always just like goofing around. We have like a little comic saying like how like we distract each other so much, we never get any work done. And sometimes it's true, but so I think it works out very well. Where like with my lab mates, other grad students, postdocs in the lab, like I have kind of like more like a friendship type of relationship. With my PI, it's like a very supportive professional like mentorship, and I really value that. So it's a good fit for me, right? But not for everyone.
Also, please don't listen. Not to throw any shade at students or graduate students, but don't pay too much attention to. Don't work with that PI just because, like you say, test it for yourself. If the research is interesting, if you feel like that's something that you can move forward, that's something that you're gonna be like okay doing, test it before you decide that it's not yours. Then maybe that's why you have rotations, right? Yeah.
I agree.
I have something really quick to add. I think it's really important amongst underrepresented groups to have a lot of like combo time. in lab spaces, because I think a lot of times things get lost in just communication, because different groups communicate in different ways. And I've noticed that it's been really beneficial to be able to have open conversation about projects, life, for extended periods of time whenever you can. So just having an open line of communication that, hey, it might be a 15-minute conversation or it might be an hour conversation until we get to a resolution, or like we see on the same, like we're talking about the same thing, but we're talking about it from two different ends sometimes. And that's just a communication barrier that I think often happens. So I think I would, if you're a grad student, I think that it's great for you to advocate for having that open line of communication. You might not always need it, but when you do need it, you really do need to communicate about a project just to get as many words out there until you see eye to eye. But if you're a PI, I think that's also a great environment to create.
One thing you just triggered. I think, yeah, there's no such thing as a good PI or a bad PI. But I think one quality that I think is very, very important to look for is for a PI with good managerial skills. I think over the course of a PhD, like a postdoc that's years long, there's going to be conflict with your lab mates, right? For example, and you want a PI that's going to be able to handle that situation. It shouldn't fall on you, right? So I would say that's one quality that I think it's pretty universal to like a quote unquote good PI.
Any other questions? Yep.
So it's actually following up on that. So Tere Padilla from Wesleyan. Uh, Academia is a learning process that will end the day we die, period, right? We are always learning, like I'm learning from you, from postdoc, from my students, right, all the time. When I was getting my PhD several years ago, let's not get into the numbers, I got my PhD in Mexico. It's a completely different environment, a completely different atmosphere that's been changing over the years. When I came to the States, it was a scientific cultural shock, but like I think I adapted and I survived. I'm here. But like my question, thinking on this changing of generations and also cultural competency and whatnot, what you guys think now in your different positions is the most challenging and difficult conversations to have with your PIs? Like thinking, like I like to have an open lab, safe environment. That's I strive for that. Sometimes it's hard, right?
Right.
There's a rotten apple. Things can go wrong, right? But what for you guys, in your spot, is the most challenging, hard conversation you may have with your advisors? Jen.
So I wouldn't say that it's a hard conversation. I would say it's an uncomfortable, maybe a slightly awkward conversation. But when I first got into the lab, like just even coming into graduate school to begin with, I was always told, be careful whose lab you join. Some people just want you because they may get extra funding for you. Some people just want you so that you can do all the work. And so I had a conversation with my advisor on just this notion of trust and what were her interests in me. Did she think that I would be a good scientist, or was she just taking me in for the work that she hoped I could do? Like, um, it was a very awkward, uncomfortable conversation, but I felt like it was one that I needed to have with her so that I knew where we stood, because I'd also known PIs who didn't think I deserved to be in that space. So for me, it was more of like how do I survive here? I need to know your thoughts on me.
So, and to add on to my question, like, how, in your opinion, make this better? Like, you being the mentor, how would you fix that? Like, you have this problem, like, it's challenging for you to talk about it. How you being the PI would help that person to be better or do better?
I would say that my advisor, she showed me that she really did value me. She showed me that she trusted me. And it also helped that, like, I could I don't know, we're in a group setting and I could overhear her talking about me and saying that, oh my gosh, Letitia is amazing. She does this, 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 and this. Right? So even though she didn't know that I was listening, she was still talking positively about me. So for me, that's like what allowed for that trust, and that, like, okay, she does have my best interest in mind.
Thank you.
Yeah, this is a very difficult conversation, especially I think you're hinting at something that affects a lot of underrepresented minorities, which is the diversity supplement, where some students, and I have had students that have actually been invited to labs because they check the minority box, not necessarily for any other reason. And it shouldn't be that way, but those are things that do exist. I think as a majority person, maybe you're interested in having a minority trainee. I think the main thing is that we're all saying is this open line of communication. I think that when you can talk openly and make it comfortable to say, hey, I believe in you as a student, as a trainee, as someone that's going to do well, then it builds confidence in the trainee. And then they can know that this is a person that I can be successful with. And I think that that's what's happened for me in a lot of ways is I've had people that have invested in me. And I think investment from a PI shows a lot to the trainee that this is someone they're willing to support. And then when they start to open their network to you, there's certain ways that PIs kind of spread their wings to their trainees, right? So if I were to become a PI, I think my main way would be to leverage my network for them, making sure that they are invested in. And it comes with the currency, right? Are they getting papers so that they can be successful in their next step? Can they get a good postdoc because they came out with all of these publications? I think that I was invested in very well as a grad student. And so it set me up for a lot of competitive postdocs so that when I got there, it was like, well, she came in with 12 papers. Let's elevate that, right? So those are things that the PI did to support me, and that is something that I will continue. But those are difficult conversations. There's no right or wrong way to actually navigate that conversation. But I think making sure that a trainee at any level, undergraduate up to postdoc level, really feels included in the conversation and really feels that you are have their best interests at heart and not just your own political or scientific interest at heart.
So the most difficult question I mean, conversation I had with my PI is right there.
Dun, dun,
Uh,
dun.
I think that way. I mean, I'm not going to talk about what we've been talking about in private, but the most difficult conversations were when I've been feeling that he wasn't advocating for me. And to be fair, as you take time to think about it, I will join Mars by saying that it was more of communication barrier, the fact that we are coming from different cultural background, that what maybe made the conversation difficult. And I think when two stubborn people are talking, the conversation can really get derailed and they can forget uh, what the most important things are. So something that I'll be working on by myself, and if I become a peer, that's what I will continue to work on, is really understand that not everyone is necessarily going to understand things the way that I do, and sometimes it's going to be important for me to navigate and to make sure that I understand what the student wants to say, even if the student is not using the same words that I'm using. And me as a graduate student, I'm also trying my best to not just necessarily think that the system is against me, and that's because we all grew up in the system, and just as we have been influenced by the system, everybody has also been influenced by the system. Not saying that it happened the same way for everybody, but sometimes someone might be doing something not knowing that what he's doing or what she's doing is hurting someone else. So not always thinking that that person is against you or that person did that on purpose, but just trying to communicate what you said again, continuing to communicate until until you feel like the older person understood what you wanted to say. But I think it's also important to know when to say okay, let's stop it here, let's take a break, and let's continue later. And for example, me, something that I've started doing is when something happened that I feel like I really need to say something about it, I don't just get worked up right away. Usually they say, okay, let me take 24 hours, 48 hours to think about it before I bring the conversation up. So, yeah, as a PI, I think if one of my students were to have, or one of my trainees were to have one of those difficult, I would encourage them to take time to think about it first. Maybe write some ideas down, like go through the critical thinking, and then just try to have many conversations about it, and try to make each other understand that we are not necessarily working against each other. We are working toward the same goal, but it's just that we might be doing it differently.
To the last part of your question, like what can you do as a mentor to make somebody feel more comfortable? I immediately thought to this one. So I joined my lab first as a post-bac student. I did a prep program, which if anybody in undergrad is thinking about PhD and they still need a little more time to think about it, I would consider a prep program. So I joined my lab as a post-bac student with no background in immunology into the like very hardcore immunology lab. So I remember I was like very like everything was going over my head for the first few months in the lab, and I remember very clearly one conversation I had with my PI about where we were talking about the project, and he turns to me, he's like, "Well, what do you think?" And then I was like, wow, like it's the first time that you've asked for my input, and that just felt very, very like comforting. I was like, wow, like my opinion matters here. And I think from that point forward, it just became so much easier for me to like ask questions, like in like lab meetings or when I don't know something, instead of pretending like I didn't know something, I was like much more comfortable being like, oh yeah, can you tell me a little bit more about this? So I think like something as small as like asking like for their input can go a long way. So.
Thank you. Yeah, sure. Go ahead.
Hi. Oh, hi. I just had one question. So this is for everybody up there if you want to answer. But I know like grad school can be like stressful and hard, and it's a lot of pressure to be put on you. What are just some things that you do to keep you going, or just to keep you leveled while you're in school, to keep you level minded, so you don't like burn out or anything?
I'd say real quick, pick the institution, knowing what options you have. Don't sign up for a 6 year commitment and not have been to the city or know what outlets are already there. Providence, I picked because it's on water, and water's my peace. So like going to Newport or just sitting in the bay on the bridge, that's always a great way for me to release at least.
I guess for me, it was having an outline of what my greater goals were. And so, one example of that is I actually found out about the HHMI Gilliam Fellowship when I was an undergrad, and I found out when I was a sophomore. And it was a goal of mine when I was in grad school that at the very least I would apply. And it ended up coming to fruition, and now I'm a fellow. And so I think when it comes down to knowing where you want to go and knowing what you want to see in yourself, and maybe you don't know right now, but think of someone that you admire. For example, Dr. Extavor, like how do you get to those positions, and how can you be the best scientist, your best mentor, the best teacher that you want to be. So yeah, I think like writing down what you want to do and seeing the plan to get there.
Personally, I think I would just say leave campus whenever you can. I think it can be a very, I guess, claustrophobic environment. Like thinking, like there's times where I'm like dreaming about lab, and I'm like, like gotta take a break from that. So I would just say like find the things that you enjoy, the hobbies that you like, the people that you like that are not scientists. I think those are very important people in our lives to just like unwind and have like a little outlet. Like for me, that's like soccer. I love playing soccer in like evenings with like some of my closest friends now, and it's just like a time to just shut off your brain and like like unwind.
My go-to is yoga. Uh, like I set up my schedule to where every single day at the end of the day at 5:30, I had a yoga class to teach. And so because I had to teach it, it forced me to be accountable because if I wasn't teaching it, I wasn't gonna go. And then on top of that, I think more so like a routine is what kept me going. Like regardless of what happened during the day, I knew that my morning would start off with a cup of tea and looking out my window at the mountains, and I knew that the evening would end with a yoga class. And so knowing that those two things would be constants regardless of what happened, that kept me going.
I love that. So let's see. I think I've set up small wins for myself. I celebrate everything, the small ones, the big ones, all the things that happen. And I think what motivates me is feedback. when I get feedback from my peers, when I get feedback from random people on Twitter, like, oh my gosh, girl, I love that. That's a great, or I loved your post, it inspired me today on Instagram or something. Feedback lets me know that even if it's just one person, that something I said resonated, that impacted their day. And then I feel like my purpose has been fulfilled. So I'm very much purpose driven. What am I doing? Is it impactful? Is it reaching any persons? Is it doing anything? And that's kind of how I use to motivate myself. And then just spending time with family. I think family is super important to me, but they motivate me to keep going.
Also, nothing lasts forever. Meaning that even if you are stressed in grad school, it doesn't last forever. Something that I always like to tell myself is, in 5 years it won't happen anymore, or in 5 years, in in 5 years I would think back at this situation and just laugh. But I would second the fact that every day is a new day. Every day is a chance to make yesterday a little less hard. And for me, usually, I like the fact that things have deadline, like I said, and um, yeah, basically just that. Things have deadline. It's hard. Find the things that allow you to get up the next day to push forward. Keep your eyes on the goal, but don't forget that in order for you to go to the goal, you have to step on every steps on the staircase. But other than that, it's just basically what we've been saying all throughout. Surround yourself with people that make it worth it. Surround yourself with people that you can go to the bar with after a really hard class and talk bad things about the teachers and PIs behind the back. Uh, and just feeling better overall. Yeah, yeah, and just your cohorts, your other graduate students, you're not going through it alone. Sometimes you feel like you are going through it alone. You're not alone in it. All of us, regardless of our race, again, are going through the same thing because our race is graduate student, our race is not all the other things. We have that on the side, but that's who we are. And most of us have the same issues. Just don't close up to yourself. Like I said, I have those two people that I know that no matter what happened, they understand. And yeah.
Well, thank you everyone for joining us for this discussion. I think we had a phenomenal array of topics to think about for starting on a successful path, especially for the trainees. And let's please give a round of applause once again to all the panelists. And now we're going to turn it over to Dr. DeLong for the closing.
Before I start thanking people, I have two important pieces of work to do. So one piece of work is to remind you that we have a reception waiting for you outside, which you have richly earned at this point. And this has been put together by Miles Mundy, who unfortunately is ill and couldn't be here to enjoy the reception himself, which is really terrible, but also Carolina Mejia Pena and Samara Cummings, who I think are waiting for us outside. And the reception really is focused on local specialties that are produced by local BIPOC businesses. So I hope everybody has a great time. There's a real range of food and things to enjoy outside. I also want to remind people we have some lovely stickers. That's our swag. And I would encourage you to pick one up. They're on the table next to the elevator here. You can put one on your phone or your water bottle or your computer or your office window, whatever you like. And then the next piece of business that I should do is to tell you that next year's meeting will be June 6th and 7th. So essentially this same Thursday, Friday window, 2024. And we're delighted to announce that our keynote speakers are going to be Dr. Sherry Lynn Black from Duke and Dr. Blanton Tolbert from HHMI. And at that point, he will have moved to the University of Pennsylvania. So I hope that all of you will consider coming, and I hope it'll work in your calendars. We would love to see you again. And so with that, I really have a bunch of thank yous to give. I want to thank all of the members of the committee, and I also want to thank the media services crew who have really supported us excellently, and Connor and Alex and Laura making everything work for us, which has been really great. I want to thank Amy Cohen, who I know is lurking over there on the sideline, so maybe this time we can clap for Amy when she's here.